We had an exciting session today talking really about the boundaries of immunotherapy now in cancer and its very rapid pace of development. And my specific topic was on CAR T cells that have just recently become FDA approved and their uses in leukemia and lymphoma. And then I spent some time discussing the use in experimental settings such as solid tumors like prostate cancer. Yeah, so and this is a concept that drugs are purchased and they're given one time, and then they metabolize and leave really no trace in the patient. CAR T cells, what I mean by process, is similar to a bone marrow transplant. In a bone marrow transplant, one finds a donor and then basically exchanges the immune system, and that requires a whole team of patients or physicians and a specialized treatment. So CAR T cells, it's, the patient themselves is a donor, but it requires manufacturing, and then when the cells are given, they stay there, we now know, for up to 7 years in the patient. So it's a living drug, unlike the standard drugs that are metabolized. Yeah, the first patient we treated was in 2012, and that was Emily Whitehead, and she was reported in the scientific literature a year later, and she's now more than 5 years out without any further therapy. So she was a striking result. When that happened, though, she almost died of cytokine release syndrome. In the publication, in the New England Journal of Medicine, we showed that her fever was 106 degrees for 3 days. So she had a very severe reaction, but that correlated with the elimination of her tumor. So we didn't know when we first treated her if that was a fluke, and she was going to be a rarity or not. And it turned out our first 12 patients, we had a 92% complete remission rate. So it was striking how well it worked. And that became FDA approved for people with acute leukemia ages 3 to 25 in the United States last summer. Yeah, so cytokine release syndrome is the fever that occurs when the tumor is being attacked by CAR T cells. And we do know that the chance of getting cytokine release syndrome depends on how much tumor you have. So if you have a packed bone marrow with lots of tumor, then about 50% of the patients get cytokine and end up in the intensive care unit. But it also correlates with efficacy, so that if the patient doesn't have cytokine release syndrome, they're usually the ones that don't respond. And the primary cytokine that causes the fever and the cytokine release syndrome, which can lead to other organ system failure if not treated, is called interleukin-6, a cytokine that now can be blocked by a drug called tocilizumab, which is an IL-6 receptor antagonist. And that was co-labeled with CAR T cells that are now marketed both by Kite and Novartis. Yes, and so CAR T cells appear to combine well with many other modalities, such as checkpoint therapies, oncolytic viral therapies, and some targeted therapeutics, such as ibrutinib. So we think there'll be many combinations. One limitation right now is that the preclinical models are not very good at telling us what is going to be the best combination, or whether, and sometimes there might be antagonistic effects. Yeah, so at this point, the big scientific challenge to the field is developing CAR cells for solid cancer. And so far the results have been generally disappointing, and I think we now know why. And one issue is that the CAR T cells targeting solid cancers have had a paucity really of good targets where we could not have lethal on-target effects but off the tumor. So for instance, causing encephalitis at the same time the tumor goes away, or lung reactions at the same time the tumor, if there's shared antigens. So the optimistic aspect, though, is that if we take tumor biopsies from a number of adenocarcinomas. And in our case, we've done studies in ovarian cancer, pancreatic cancer, and lung cancer. We find that in a petri dish, the CAR T cells do kill the cancer cells, and in a very similar way that they do in leukemia. So I think the optimistic aspect is the CAR T cells can kill, and it's going to be an issue now of solving the targeting issue, which can be done with CARs that have multiple targeting warheads, we call them, and then as well as being able to shut them on or off. And I think all those things are in a laboratory at this point. So yeah, when we infuse CARs, they actually expand, we in some patients more than 100,000-fold from what we infuse. So there's the manufacturing we do in a laboratory, and then that in the patient. And it will obviously become cheaper if we can make most of the manufacturing, meaning growth of the CAR T cells, happen in the patient rather than the laboratory. So it's very different than other drugs where there's a fixed dose and then they're metabolized. In this case, we actually need less CAR T cells with the patients who have the higher tumor burdens. It's very counterintuitive, but it turns out the higher the tumor burden, then that drives more proliferation of the CAR T cells, so we need less. And so it's very different than standard pharmacology when you have a living drug that reproduces in the patient. I think that, in fact, is quite likely that, because we know when you do a bone marrow transplant, if you give, there's both stem cells and then the passenger T cells are called. So if you had someone who's been a CAR T cell recipient and then they become a bone marrow donor, potentially the CAR T cells could be transferred from the original patient into the recipient, and then they would take root, and that patient would then also have CAR T cells without ever having gone through the process. So one thing we do look at, so that would be an unusual case, but I think that's more than theoretically possible. I think it would happen. But what is a more severe issue is if a patient's, we use engineered viruses such as lentivirus or gamma retroviruses to introduce the CAR T cells. And it's in the past, with less sophisticated methods, it was possible that those viruses could become replication competent and then that way infect other cells in the body. So, and if that would happen, then that could be very serious if the patient had replication competent virus that's used to make the CAR T cells. That's never happened at this point, but it's being watched by all the FDA and other health authorities.